In a lysogenic cycle, viral nucleic acid becomes part of the host cell's chromosome and is replicated with it. Eventually, the virus enters a lytic cycle and kills the host cells. A bacteriophage attaches to a bacterial host cell by locking onto a specific receptor site on the surface of the host cell. The virus then injects its DNA into the host cell. Once inside the cell, the viral DNA inserts itself into a specific site in the chromosome of the host cell. Viral DNA is then called a provirus. During cell reproduction, the host cell copies the provirus genes along with its own DNA. The provirus is inactive at this time. When the cell divides, both the host genes and the provirus genes are passed on to the two daughter cells. At any time, a provirus may leave the host chromosome and enter a lytic cycle. When this happens, the host cell's DNA is broken down, and new virus parts are produced and assembled. Eventually, the cell ruptures, and new virus particles are released. Is a viral reproductive cycle in which a virus takes over all metabolic activities of a cell, replicates itself many times, then destroys the host cell. A bacteriophage attaches to a bacterial host cell by recognizing and locking onto a specific receptor site on the surface of the host cell. The virus then injects its DNA into the host cell. The empty coat remains outside the cell. Inside the cell, the viral DNA breaks down the host cell's DNA. The virus then takes over the total metabolic activities of the host cell. By using the raw materials present in the cell, the viral DNA directs the production of new virus parts. The newly produced viral components are assembled into complete new virus particles. The host cell bursts open and releases 100 to 200 new virus particles. These new particles can begin another cycle by infecting nearby cells.
Targeting HIV replication. The replication of HIV-1 is a multi-stage process. Each step is crucial to successful replication and is therefore a potential target of antiretroviral drugs. Step 1 is the infection of a suitable host cell, such as a CD4-positive T lymphocyte. Entry of HIV into the cell requires the presence of certain receptors on the cell surface. CD4 receptors and co-receptors such as CCR5 or CXCR4. These receptors interact with protein complexes, which are embedded in the viral envelope. These complexes are composed of two glycoproteins, an extracellular GP120 and a transmembrane GP41. When HIV approaches a target cell, GP120 binds to the CD4 receptors. This process is termed attachment. It promotes further binding to a co-receptor. Co-receptor binding results in a conformational change in GP120. This allows GP41 to unfold and insert its hydrophobic terminus into the cell membrane. GP41 then folds back on itself. This draws the virus towards the cell and facilitates the fusion of their membranes. The viral nucleocapsid enters the host cell and breaks open, releasing 2 viral RNA strands and 3 essential replication enzymes: integrase, protease, and reverse transcriptase. Reverse transcriptase begins the reverse transcription of viral RNA. It has 2 catalytic domains: the ribonuclease H active site and the polymerase active site. Here, single-stranded viral RNA is transcribed into an RNA-DNA double helix. Ribonuclease H breaks down the RNA. The polymerase then completes the remaining DNA strand to form a DNA double helix. Now, integrase goes into action. It cleaves a dinucleotide from each 3' end of the DNA, creating 2 sticky ends. Integrase then transfers the DNA into the cell nucleus and facilitates its integration into the host cell genome. The host cell genome now contains the genetic information of HIV. Activation of the cell induces transcription of proviral DNA into messenger RNA. The viral messenger RNA migrates into the cytoplasm, where building blocks for a new virus are synthesized. Some of them have to be processed by the viral protease. Protease cleaves longer proteins into smaller core proteins. This step is crucial to create an infectious virus. 2 viral RNA strands and the replication enzymes then come together and core proteins assemble around them, forming the capsid. This immature viral particle leaves the cell, acquiring a new envelope of host and viral proteins. The virus matures and becomes ready to infect other cells. HIV replicates billions of times per day, destroying the host's immune cells and eventually causing disease progression. Drugs which interfere with the key steps of viral replication can stop this fatal process. Entry into the host cell can be blocked by fusion inhibitors, for example. Inhibition of reverse transcriptase by nucleoside inhibitors or by non-nucleoside reverse transcriptase inhibitors is part of standard antiretroviral regimens. The action of integrase can be blocked. Protease inhibitors are also part of standard antiretroviral therapy. Each blocked step in viral replication is a step towards better control of HIV disease.
If you haven't seen our previous video about proteins, and you want to be extremely wonderful, I suggest you watch that video before this one. It'll definitely help you understand prions and proteins much better. Proteins are obviously extremely important to our everyday life, but what happens when these proteins decide to become a killer? Some proteins, even though extremely rare, can misfold from these really weird proteins called prions. These proteins actually misfold other properly folded proteins, causing an exponential chain reaction of misfolded proteins, and they all end up binding together. In an infected person, these proteins are located in the brain and cause neural issues and death quite quickly. These neurological disorders are known as transmissible spongiform encephalopathies, or TSEs for short. They are a group of the rarest diseases and have the highest mortality rate on the planet, with 100%. There are no known cures and no treatments available. TSEs are extremely scary. Did I mention that these proteins can't be denatured by proteases, enzymes, heat, radiation, and even autoclaves, which heat up to 134 degrees Celsius in a pressurized steam chamber for 18 minutes and are meant to kill any and all bacteria and viruses? So now that I've scared you, how do you get this horrific disease? It's quite complicated, and of course more research needs to go into it, but there are several ways we think we can get it. It starts off knowing that there is a cow version of the disease. You've probably heard of it, mad cow disease. If humans eat beef infected with the prions, and the prion makes it to the brain, it can start causing the disease. It's also thought that if humans come in contact with manure or urine from the infected cows, they have a chance of being infected with the prions. The cows get it the same way humans do, by eating infected meat. The good news is that the US banned the practice of feeding cattle other cow parts in 1997. Kuru is another version in which human cannibalism can cause it. As gross as this might sound, it's mostly the people who eat the brain. There's also an even rarer genetic version. Personally, I think the genetic version is by far the scariest. It causes progressively worsening insomnia, which leads to delirium and a confused state of dementia. You get so sleepy that you go crazy and eventually die. Luckily, TSEs are extremely rare and only affect about 1 out of every million people each year. Even though prions and the diseases they cause are extremely scary, we have a lot of preventative measures to keep them hopefully out of the population. More research needs to be done on the prions to understand the ways to combat them and possible treatments for the issues they cause. Just be glad that your proteins didn't decide to go rogue.
There is evidence that the infectious agent associated with certain brain diseases, such as Creutzfeldt-Jakob disease in humans and mad cow disease in animals, may be caused by a protein. The infectious agent has been called a prion, for proteinaceous infectious particle. The normal prion protein, PRPC, is bound to the surfaces of neurons, and the gene for PRP is present in normal vertebrates and invertebrates. Presumably, the normal prion protein, PRPC, can be altered, and the altered prion protein, PRPSC, SC meaning scrapie associated, becomes misfolded and takes on a different conformation. Many researchers believe that the infective agent is this alternative protein conformation. It is believed that when PRPSC enters a normal brain, it binds to the normal cellular PRP, PRPC, and induces it to fold into the abnormal conformation. The newly produced PRPSC molecule may then convert more normal PRPC proteins into the abnormal PRPSC form. An alternative hypothesis is that PRPSC could activate enzymes that modify the PRPC structure, not shown.